All right, in this video, we're going to discuss some details about the polymerase chain reaction, or PCR. Now, it's absolutely essential that you watch some of the videos that I've included in Canvas that animate the polymerase chain reaction. It's much easier to understand what's going on in animated videos than it is in my static images here. Watch as many videos on the polymerase chain reaction as you can. It's a deceptively complex reaction. It takes some time to really fully understand what's going on. But in my video here, I'm going to draw attention to some details that are often overlooked in other videos. Now, the polymerase chain reaction was developed in 1985 by Kary Mullis. He ended up winning the Nobel Prize for Chemistry in 1993. And it is a way to amplify, and when we use the word amplify in this particular context, what that means is it makes lots and lots of copies of a very specific sequence of DNA, fragment of DNA from a template. So this double-stranded DNA that you see here is called our template DNA, the DNA that we start with. This would be the DNA that you managed to get from a hair follicle cell at a crime scene, or the amount of DNA you're able to purify from a cheek swab. You know, if you're a criminal, or if you're a detective and you have a suspect give you a little cheek swab, well, you purify DNA from that, but that's way too small amount of DNA to do anything with. And so what you need to do is you need to perform polymerase chain reaction on that little teeny tiny bit of DNA to make lots and lots of copies. Now, you're not making lots and lots of copies of the entire DNA sequences that you've purified. You're only making lots and lots of copies of a very specific portion of that DNA. Now, how do you direct this PCR to make lots of copies of a specific sequence? You have to make primers that flank that particular fragment of DNA that you want to purify. Now, those primers are also going to become permanent parts of the fragments that you generate. Where do you get the primers? You write up the sequence online, you send it off to a company, they chemically synthesize those primers, and they send it back to you. So that's not a problem. Okay, so what is involved in a PCR reaction? Well, you need your template DNA. And the strength here is that template DNA can be a very, very, very small quantity. Then you need your primers, right? You get a whole bunch of primers and throw them in there, right? You're limited by how many primers you have. The number of primers that you add, right, is going to determine the maximum number of fragments that you can make, because each primer that you use gets used up and becomes part of the fragment that gets amplified. You know, since you're building lots and lots of DNA, you're going to need lots and lots of DATPs, DGTPs, DTTPs, and DCTPs. And finally, you need a DNA polymerase that is going to catalyze the reactions of building DNA. Now, the DNA polymerase that we use is called Thermus aquaticus, is from an organism called Thermus aquaticus. This is a prokaryotic cell that lives at high temperatures, and so its DNA polymerase has evolved to withstand the ability to be put at high temperatures without being denatured. Now, the primers. Now, this is one of the things that many of the videos you watch will sort of overlook, but I want you to be able to, if given a sequence of DNA, to be able to build primers that will amplify that DNA. So let's say we have, this is our template DNA. In the box here, that's the fragment that we want to amplify. And so what you do is you look at the 5' to 3' direction of one strand, 5' to 3', and 5' to 3' of the other strand that go in the opposite direction, that flank the, that match the ends of the fragment that you're trying to amplify. So in that case, here's 5' to 3'. Here's 5' in red to 3', right? Those are the opposite ends of this fragment that we're trying to amplify. And then you just take the first 20 to 25 bases. So 1, 2, 3, 4. So basically these bunch here and these bunch here. And the reason you do it that way is when you choose those sequences, they will be reverse complementary to the other strand. Boop, boop, boop. Reverse complementary to the other strand. And therefore, they will act as primers on which DNA polymerase will extend from. Now, one important thing is these primers have to have about equal melting temperatures. I'll let you think about that for a moment and we'll come back to it a little bit later. So you can't just make anything. You can't just do any sequence here. You want to look for primers that are about equal melting temperatures. All right, then you run it through PCR. So now this is where the videos come into play very well, but this also is going to be the single slide here that you're going to want to keep coming back to, that I'm going to go over some of the details. You're going to take your thermal cycler, or more commonly called a PCR machine, and it's a machine that you can program to heat up, to cool down in various cycles. And so the cycle that you want to do is the first thing you want to program in the machine is the first cycle is going to be to go to about 94 degrees. Now ignore that 5 minutes, nobody does it for 5 minutes anymore. 30 seconds to a minute is fine. And it's this first cycle that's called your denature cycle. And what it's doing is it's denaturing all of the template DNA. It's denaturing all of the fragments of DNA that you've already made. And so everything now is single-stranded. And then that thermal cycler immediately goes down to what's called the annealing step. Now that's going to be between 50 and 60 degrees Celsius. And it's really important to understand that I can't tell you exactly what temperature to go to because this temperature is dependent upon the melting temperature of your primers. Right? You want just right around the same melting temperature so those primers will bind, but they won't bind non-specifically. If you lower the annealing temperature too much, then your primers are going to start binding to sites they should not bind to. If you have your annealing temperature too far above the melting temperature of the primers, then they won't bind to anything. So you need that annealing temperature right around the melting temperature of the primers. And you can't have two different annealing temperatures. You can only have one annealing temperature, which means you got to make sure that the primers you design are of about equal melting temperatures, because you can only have one annealing temperature in your thermal cycle program. So that's really important. In addition, you don't want your primers full of repeats. You don't want a primer that's AT, 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 AT. Why? Because there's lots of sequences within the genome that are AT, 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 which means there's going to be lots of places that primer can bind, and you don't want that. You want your primer to bind a single location within the template DNA. Okay, so the annealing step. 50 to 60 degrees Celsius. Why can't I give you a temperature? Because I don't know what the melting temperature of your primers are, right? Depends upon that. Now, that'll go for about a minute. Again, these numbers are higher than what they are nowadays, so about a minute or so. Give time for all of your primers to anneal to whatever sequences they bind to. And then you go to the extension step. Now, 72 degrees Celsius is the temperature at which Taq polymerase, so Thermus Aquatis DNA polymerase, works the best. Now, why? Here's another question. Why have I given you a range of time, 2 to 5 minutes? Well, the reason is because that Thermus Aquaticus, that Taq polymerase, extends at a very specific rate, adding bases at a very specific rate. So we have to give it enough time to amplify our entire fragment. How much time is that? Well, it depends on the size of your fragment. Usually it's about 1 kilobase per minute. So you give it however much time you need. It depends upon how large your fragment is that you're amplifying. And then it starts the whole thing over again. Boom. Denature everything, including the DNA that you just created. Denature it. Boom. Go to anneal. Allow those primers to anneal to their target sequences. And then again, step 3, extend. make new DNA. And you do this over and over and over again, and it's usually a PCR reaction tends to be about 30 to 35 cycles, and at that point you have lots and lots of DNA. Okay, we don't need to see that, and so just here's a kind of a rundown of the first several cycles of PCR amplification. Now, since the advent of the polymerase chain reaction, there are all sorts of variations, many of which are going to be used in the paper that you guys are reading, so you have to understand this. The first is that we can use PCR as a way to quantify just how much DNA we started with. So just how much DNA were you able to purify from those few blood cells you found at a crime scene? How much DNA were you able to purify from that hair follicle cell, or in a single cell that you got from Drosophila that you're studying? Okay, this is a very sensitive quantification technique. And how does it work? Well, there's a molecule called cyber green, and that molecule, when not bound to double-strand DNA, does not fluoresce. It only fluoresces when it's bound to double-stranded DNA. So what you do is you run a PCR reaction in the presence of this cyber green, and after every cycle 1, cycle 2, right? Step 1, 2, and 3. Cycle 2, step 1, 2, and 3. Cycle 3, step 1, 2, and 3. After every cycle, you put your reaction is then put into a spectrophotometer, and the fluorescence is measured. Okay? This is called real-time PCR, and you have to be using a special PCR machine to do this. And what you're doing is you're looking for at what cycle the fluorescence passes or reaches this threshold fluorescence. And depending upon what cycle that threshold was reached will determine how much DNA you started with. Now, the cycle at which the threshold fluorescence was met is called CT, cycle threshold. And the earlier you've reached that threshold means the more template DNA you started with. So if we take a look here, now you can take a look. Okay, well, we've got this standard curve. Well, my CT, the cycle at which my threshold was reached was at 15, very early. So how much starting DNA did I have? Boop, 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 boop. Well, I had about 10 to the 4 micrograms or nanograms of DNA. Notice, the bigger your CT, the less amount of DNA that you started with. Okay, so make sure you understand what CT is and how it's relating to your starting DNA concentration. Now, that's called qPCR. Now, another type of PCR is called reverse transcriptase, or RT-PCR. This is a way that you can do two things. You can identify particular RNA molecules that are in one of your samples or in one of your cells, and you can quantify how much RNA was in your sample or in one of your cells. And so what you do is you have your RNA sample that you've purified, and you have a primer that is reverse complementary to the 3' end of your RNA that you're trying to measure, that you're trying to amplify. And you run a reverse transcriptase reaction, which means you're reading RNA, but you're writing DNA. So at this point, you've essentially made half of a cDNA. And then into that mixture, you throw in a second primer. Now that primer is the pair of the second part of the pair of a PCR reaction. That's going to bind to the other end of this DNA fragment that you've just created. Now you're going to put your Taq polymerase in there. Now you're going to throw that whole mix into a PCR machine, and you can then amplify that cDNA that you've just created. So this is probably a more easy way of creating cDNA than what you what I taught you before. The problem is, and the difference is, this one here, you're creating a very specific cDNA. In the other technique that I showed you, you're creating lots and lots of cDNAs at the same time. Now, if you threw cyber green into the mix here, then what you would have is RT-qPCR, so reverse transcriptase quantitative PCR. So you can combine some of these techniques. Now, this is going to be a technique that's going to be extremely common in the paper that you guys are reading. Let's look at a really neat use of PCR, and it's called DNA genotyping. Sometimes it's called DNA fingerprinting, DNA profiling. And what this takes advantage of is the fact that 3% of our genome are simple sequence repeats. What do they look like? Here's on chromosome 7 of humans. This is the D7S820 repeat. Notice AT, 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 AT. Let's see, ATAG, 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 ATAG. You get the picture. Now, what's important about these repeats is that they are polymorphic. What does that mean? It means that in different people, this stretch of repeat is different sizes. And of course, like anything else, that's passed on to the offspring. So if your mom's size of this was 150 base pairs, then you are going to inherit a 150 base pair size of this as well. Okay, so different people out there in the world have different size repeats, and it's broken down into, right, if there's not a million different size repeats here, there's probably 5 or 10 within the population. But when you look at a whole bunch of these different repeats, so we've got about 13 of these different repeats. This is CODIS, the criminal database, that anytime they take a criminal, they run and they figure out the size of repeats for each one of these alleles. And here's the repeat length. Here's the number of alleles seen in the human population. So you don't get an infinite number of different sizes, but there's quite a bit. And when they run all of this out and they look at what is the size of your repeats for each one of these alleles, they can 99.999% of the time match you to you and nobody else. Here's the question. How do they quickly determine the size of your alleles? Well, they do it via PCR. I got some questions here though. What is the max number of alleles that any one person can have? So if we have, here we have chromosome, that repeat on chromosome number 7, how many different sizes of this can an individual have? Well, remember, we are diploid organisms. That means we have 2 copies of every chromosome, which means we have 2 genes, 2 of every genes. And in regards to these repeats, we have 2 copies of each repeat, which means we get 1 from dad, we get 1 from mom, and so we can have a maximum of 2 different sizes of this repeat. And the easiest way to figure out what sizes you have is to run a PCR reaction on your DNA. So a scientist will take a little bit of swab of your from your cheek or a hair follicle or whatever source of teeny amount of DNA that we can get from you. We run that through a PCR reaction using primers that don't include this repeat. That would be terrible. If you have made a primer that included that repeat, that primer would bind all kinds of places within the genome. So it's got to be on the edge of that repeat in the genomic DNA that is itself not repeated. You take your template DNA, you take those primers, you add some dNTPs and Taq polymerase, and you run your reaction, and then you take that reaction and you run it on a gel. Right? That's the cool thing, is that this PCR reaction is so specific, and it purifies, or excuse me, it amplifies just that one fragment of DNA that you can actually see it on a gel. It's not going to look like a smear. So what we have here is this is a gel from a criminal case in which we had, let's see, the female cell. So if this was a rape case, and they took her cells and they ran a PCR reaction using primers that amplify a specific repeat, and here are her two alleles, this one and this one. Okay. Then we have two suspects. We have this suspect here, had maybe it looks like a little doublet, and then this suspect here had these two alleles. And then here is the sperm DNA that was found on the scene, and it had these two alleles here. Aha! The sperm DNA had these two alleles. That matches suspect 1. It doesn't match suspect 2. It doesn't match the boyfriend, and therefore we know who the criminal was. Now, remember, sperm cells are monoploid. So how is it that this person is having two alleles here if the sperm is monoploid and only has one copy of that allele? Well, we're looking at a pool of sperm cells, right? Not just one. And so some sperm cells have this allele, some sperm cells have that allele. PCR has a ton of uses. The fragment that you amplify can then be cut, right? If there's ECOR1 sites or some sort of restriction endonucleosites in that fragment, you can then digest it with a restriction endonuclease and then insert it into an expression vector. Boom. Now you've got yourself a way of making the protein encoded by that particular cDNA, for example. You can quickly compare sequences of different organisms because you can amplify small amounts of DNA and then sequence them. You can amplify DNA from ancient samples. So if you have ancient samples that the DNA is barely intact, it's a little teeny bit, you can amplify that DNA. You can detect viral infections before symptoms arise. In fact, PCR is one of the main ways in which we're using to detect whether someone has been infected with COVID-19 right now. Site-directed mutagenesis. This is a neat one. If you incorporate a mutation into the primer, remember those primers are going to become a part of the fragment of DNA that you make. And if you just make one little change here, that primer can still bind to the template, but now every fragment that gets made is going to have that little change. So you can incorporate mutations into fragments quite easily using PCR. All right, on our next video, we'll talk about chromatin immunoprecipitation. But for now, remember, make sure you go to videos on YouTube, go to videos that I've showed on Canvas, and watch PCR in action. It will become a little bit more clear, hopefully, because it is something that's super important, but is a little more complicated to understand than what you might first see.